Hello everyone, and we'll be starting with the discussion week 6 for the RNA biology lectures. And as you all know, we have in the last week started with the transcription in bacteria and in basically prokaryotes. And in the last class, we have discussed about the procedure for transcription in prokaryotes and regulation of transcription in prokaryotes in the form of lac operon. So these things we have discussed in the last week's class, and today we'll be starting with the tryptophan operon, that is also another kind of operon present in bacteria, and we'll be focusing on how it is regulated, followed by the lambda, bacteriophage lambda, the choice between the lytic and the lysogenic cycle, followed by we'll discuss some of the previous year questions asked in various competitive examinations. Okay, so starting with the tryptophan operon. So as the name suggests, it is linked with the production of tryptophan. So talking about tryptophan, it's a aromatic amino acid. Okay, so talking about this kind of operon, so the lac operon was the example of inducible system. That means the products were produced only when the metabolite was present in the system. Okay, here the thing is the operon will be expressed till the time the metabolite is present in the system to a particular concentration, but it will repress the operon if the tryptophan concentration is up to the mark in the system. So this type of operon is called as repressible operon. That means the product inhibits the that means product inhibits the operon that is activated. Okay, so that means this will express genes that help in production of the proper tryptophan. Okay, so talking about the basic structures that are present in the operon. So as like in the lac operon also we have the regulatory gene unit. So that is the promoter for the repressor, and this is the gene that codes for repressor. Okay, and then we have the promoter for the tryptophan operon, the operator. The operator is also like partly covered by a term called as attenuator. So this attenuator is basically the second form of regulation of the operon. Okay, and this L stands for a leader peptide. So this leader peptide basically helps in determining the concentration of tryptophan. Okay, and there are five structural genes that are basically involved in synthesis of tryptophan. Okay, so it basically converts chorismate to tryptophan. Okay, so this was the basic idea about the tryptophan operon and the genes present in that particular operon, and it is a type of repressible operon. Okay, so talking about the repressor, so we'll move on to the simple version of the regulation where we'll discuss different things. So this is the main version, and this is how the regulation looks like. So, starting with the structural genes first. So, these are the structural genes E, D, C, B, and A. So, this is the basic like formation how the structural genes are there, and like they are produced in a single mRNA levels when there is like because this is operon, so it will be in a single polycistronic mRNA. Now, at the translation level, these genes will produce different proteins. So, TRPE produce anthranilate synthase component 1, and TRPD produce anthranilate synthase component 2. So, these two are basically component of the same enzyme, that is anthranilate synthase. Okay, and then we have trypc that leads to the production or expression of N5' phosphoribosyl anthranilate isomerase indole 3 glycerol phosphate synthase. And then we have trip B and trip A that forms the tryptophan synthase beta subunit and tryptophan synthase alpha subunit respectively. So this also forms the same enzyme but different subunits. Okay. Now these three enzymes are basically involved in five reactions that leads to the production of tryptophan from chorismate. Okay, I'm not going into the details of the process that how the biochemical pathway works, but here we are concerned with these genes. Okay, now what happens when tryptophan levels are high? Okay, so when tryptophan levels are low, what will happen? There will be no repressor bound to the operator. The RNA polymerase will bind as such to the promoter, and it will function in the same way. The transcription will happen followed by the translation. Now the the operon is repressed when tryptophan is in high concentration. So now we are going to talk about the simple version of the regulation that takes place. So now when tryptophan is present, then we studied about the tryptophan repressor. Okay. So it basically synthesizes tryptophan repressor protein that works as a dimer. Okay, so this is the binding site for tryptophan. When tryptophan is there, it will reversibly bind to the repressor molecule, and that will activate this particular repressor and directly bind to the operator molecule. Okay, thus inhibiting the RNA polymerase to transcribe the operon system, tryptophan operon system. Okay, so this was the simple version where only the repressor works. Okay, now comes the function of the leader and the attenuator. Okay, so this is basically the mRNA of the leader sequence or the attenuator. So when the tryptophan is high, so this is the main important thing that needs to be understood. Okay, so first we'll see when there is a low tryptophan. Okay, so then what will happen? The RNA polymerase can synthesize the whole mRNA of this particular operon. But when there is high tryptophan, then only the leader mRNA is formed with the attenuator, but it will not lead to the expression transcription of the structural genes. Okay, and the length of the leader mRNA is around 140 nucleotide. Okay, that is too less. So when their tryptophan amount is high in bacteria, so in that case, this leader mRNA is only formed and not the mRNA for these structural genes. Okay, so the key point how it happens lies in the peptide sequence of the leader mRNA. Okay, so this is the operator region marked in this particular green. Okay, followed by the leader mRNA. So from here it comes the leader mRNA. Okay, so this region is the leader sequence. This is in some books it is also written as strip L. Okay, so it codes for a leader peptide. So till here it's the leader peptide followed by a stop codon. Then comes the attenuator sequence. So from this green to this green is the attenuator sequence. Okay, sorry. From this green to this here is the attenuator sequence, attenuator mRNA. Followed by, from here starts the structural gene. So here it is written trip E. So here, from here, the mRNA for structural gene starts. Okay. Now, as you can see, this leader peptide, okay, it's about it's about 15 amino acid long sequence, and there are true tryptophan present here. Okay, so what happens when the tryptophan amount is high in the cell? That means this leader peptide will be easily formed. Okay, so what happens? The mRNA that is formed till here is only present. Okay, but when the tryptophan amount is less inside the cell, then this leader peptide will not be formed because the ribosome will be stuck here. And what will happen? These attenuators will form a structure so that RNA polymerase can continue the transcription. Okay, so we'll see in detail in the next slide. So for now, what happens? As you can see, there are numbers written here. Okay, these numbers represent the sequence that can pair up. Okay, so they have complementary sequences that can form hairpin loops. Okay, so this is the main thing about the attenuator. Now we'll see how this particular attenuator functions. I have told you the purpose of the leader peptide. It basically, these two tryptophan like detects the presence or absence of the tryptophan in the system. Okay, so now moving on to how this detects. Okay, so as you can see, this purple colored one has a two tryptophan codons. So when tryptophan is present in high concentration inside the cell, so these tryptophan will bind and the leader peptide is formed. Okay, and here, as you know, that in prokaryotes it's co-transcription translation, right? So here the ribosome can bind and form the leader peptide because here it's a stop codon, so it will like move on from the mRNA and it will reconnect it with the Shine-Dalgarno sequence of the structural gene. Okay, so that's a different thing. But what will happen now when the tryptophan concentration is high, this leader peptide is formed. So what will happen? These, as you know, there are four structures present here. So what will happen? These, one can bind with two, two can bind with three, and 3 can bind with 4. Okay, the structure formed between 1 and 2 and 2 and 3 are weak because the complementary base pairing between the two structures, two hairpin loops, is not that strong. Okay, but the structure that is formed by the 3 and 4, the hairpin loop formed by 3 and 4 structures or 3 and 4 sequences, is strong in nature. Okay, so as you can see in this diagram, so the 3 and 4 is a strong base pairing GC content as compared to the 2 and 3 that is majorly AU rich. Okay, that's why the hairpin loop formed from the 3 and 4 sequence is called as the attenuator because it attenuates the transcription of the tryptophan operon. Okay, so now uh, these is the main concept that works in the attenuation of the transcription of tryptophan operon. Okay, so now let's get back to the process how it happens. So tryptophan, now one thing is the leader peptide is formed in the presence of high concentration of tryptophan in the system. Now what will happen, because here the ribosome is already sitting on the first and second sequence, they cannot pair up with the third sequence. The second sequence cannot pair up with the third sequence. Okay, so what will happen, the third sequence now will pair up with the fourth sequence. So that will form a attenuator. So now what will happen, this attenuator is formed, this will not like allow the RNA polymerase to move forward. Okay, so this basically, like we have studied the row independent transcription termination. So this attenuator basically forms that only. So this leads to the termination of transcription here only. So here it is terminated because of the formation of this attenuator. Now when the tryptophan levels are low, so what will happen? This is the ribosome translating the leader peptide. So these two codons, as you know, are the detectable limit for the tryptophan. So when this tryptophan is very low in the system, so what will happen? On the first sequence itself, the ribosome will freeze, or it will not be able to move forward because the absence of the tRNA for tryptophan. So it cannot bind here. That's why this is the midway sequence of the leader peptide, and here the ribosome stops. This leads to the freeing of the second sequence so that it can pair up with the fourth sequence. Okay, now the second thing is, because the second and third sequence already has paired up, this will not be able to attenuate the RNA polymerase, and the terminator like structure, terminator like hairpin loop cannot be formed. Okay, such that the fourth sequence, that is the major attenuator, is free. So the attenuator is not formed, hence the termination is not happening and the transcription continues. Okay, so in short, what happens in the attenuation? Tryptophan levels high leads to production of the leader peptide. This leads to the formation of the loops between 3 and 4. So this leads to termination. Now, when tryptophan levels are low, so the leader peptide is not formed, then what happens? 2 and 3 forms the hairpin loop, transcription continues. Okay, so this is the whole procedure for the attenuation of tryptophan operon. Okay, now, and this leads to the end of the tryptophan operon in E. coli. So this part till here is the part for the E. coli only. Now, moving on to the tryptophan operon attenuation in Bacillus subtilis. So here there is a slight difference in like the attenuation. So in case of Bacillus subtilis, what happens? Like there were four sequences in the E. coli tryptophanoperon present in the attenuator. So like that four sequences are present, but here is the trick. The first sequence forms a upstream structure, a hairpin loop in itself. The second structure, second sequence have 11 continuing tryptophan codons. Okay, so that helps of binding a special protein that helps in termination. Okay, and this is the terminator. So like row dependent termination, this is a similar kind of process. So what happens, these yellow marked here are the continued codons for tryptophan. Okay, like there we have leader peptide to determine the tryptophan concentration. Here we have 11 codons. So now what happens when the structure follows and tryptophan is present, adequate tryptophan is present. So what happens, the protein that is trap. So trap leads stands for tryptophan RNA activated protein. So it functions as a multimer. So what will happen? It will bind to the codons of the tryptophan. Okay. So what will happen if the tryptophan is present in adequate quantity? So now what will happen? This like leader mRNA will form a leader peptide that will have 11 tryptophan molecule amino acids that will basically bind to each of the subunits of the strap protein. Okay, so now what will happen? This basically forms a snatching kind of structure so that this like this loop is not formed, and these 3 and 4 loops are formed between the 3 and 4 sequences, and a terminator is formed. Okay, so this leads to the termination of transcription of the tryptophan operon. When what happens, if there is a deficiency in the tRNA containing tryptophan, because of the tryptophan deficiency, so in that case, what happens? The anti-terminator. This 2 and 3 sequence is the anti-terminator. So this persists and 3 and 4 sequences are not formed. And what will happen? This anti-terminator basically binds to the trap protein also. So this leads to its repression and it will not be able to bind to the leader sequence. So now what will happen? Transcription will continue. Okay, and now when there is there is very less amount of tryptophan and when there is no amount of tryptophan, so what will happen in that case? Trap cannot bind to any of the tryptophan, so there is no need of the anti-terminator also. So here the anti-terminator of the transcription persists because it will like these sequence have complementarity and the transcription also continues. Okay, so similar thing happens as compared to E. coli, but the different thing is the usage of the trap protein and the 11 tryptophan molecules that helps in the attenuation process. Okay, so this leads to the end of the tryptophan operon. And if you guys have any doubt, you can ask in the comment section or in tomorrow's class. Okay, now, moving on to the other regulation of transcription, that is bacteriophage lambda, a transcriptional switch. Okay, so what happens in case of bacteriophage lambda? So, this particular bacteriophage lambda is the virus. Okay, bacteriophages are virus that affects E. coli. Okay, so it like this is the electron microscope image of the bacteriophages. So these are the bacteriophages and this is the bacteria. So these are being produced by the bacteria. Okay, so this is the major single bacteriophage. So this is the head, this is the tail, and this is how in the next slide we'll see how it will end. Okay, so it basically infects the E. coli bacteria, and this is a free phage. So it has basically tail, sorry, head, and then it has a neck and collar, and then these are the tail fibers. Okay, and the DNA is present in the head region. So it has a double stranded linear DNA kind of thing. It circularizes when it needs to be replicated. Okay, so these are the components. I'm not going to detail. Just I need to just make you understand that when it infects the bacteriophage, then what will happen? This particular DNA is introduced into the bacteria and not the whole protein. So it just introduces this particular DNA molecule, its genetic material, into the bacteria, leaving the protein molecule outside. Okay, so this was the basic thing that Hershey and Chase used to determine that DNA is the genetic material and not the RNA and other biomolecules. So that was a legendary experiment that was done. Okay, now what will happen? So it has two life cycle basically. One is the lytic phase and other is the lysogenic phase. So in simple terms, lytic phase basically stands for the production of bacteriophage by lysis of bacteria. Okay, and lysogenic means the phage genome will integrate into the bacterial genome as prophage due to unfavorable conditions for the phage to be produced, and this prophage will act like when the conditions are favorable, and it will again enter into lysis. Okay, so these are the two basic phases of the bacteriophage, that is lytic phase and the lysogenic phase. Okay, so this is the basic cycle. So when the bacteriophage infects itself, it in sends its DNA into the bacteria, and here there is a decision point. Okay, so here the transcriptional switch basically acts when it will like have to replicate in the genome of the lambda phage, and or it will integrate into the genetic material of the bacteria. Okay, so when first we'll cover the lysogenic phase and then we'll go into the lytic phase. So what happens in the lysogenic phase? It integrates into the bacterial genome as prophage, and it divides with the bacterial cell itself. So when the genome of the bacteria replicates, then only the phage will replicate. Okay, when there is certain conditions, like when UV light is induced to the bacteria, then what happens? This prophage basically comes out of the genome and it forms a circular extra chromosomal kind of genome, where it will replicate using the rolling circle replication, and then it will express the proteins that are present, that is head, tail, and collar, head, neck, and tail, and it will lead to cell lysis, and then it can reinfect the bacteria, other bacteria. So this was the lytic phase. Okay, so these were the two phases, and here how the bacteria decides where to lyse the cell or lysogenize, move into the lysogenic cycle. So that is the major transcriptional switch. Okay, that we are going to talk about. So this is the basic genome of the bacteriophage lambda. So as you can see, it has a circular genome, and starting from the cos sites. Okay, so this is the end of the bacteriophage genome. That, so it's a linear genome. Okay, it ends are cohesive in nature. That means this has complementary sequence with this particular end. Okay, so this cos means cohesive. Okay, so this has complementarity with this particular sequence so that they can circularize while replication. So this is the thing that happens while replication. This cos site is formed. Okay, now it has head genes. So these particular genes code for proteins involved in head synthesis, that is this kind of structure. Okay, the tail genes As you have seen in that, it produces tail kind of structure that helps in adherence to the bacterial cell. Now comes the various genes. These particular genes, okay, S and R are involved in lysis. Okay, Q is basically involved in late transcription. That means the proteins involved in synthesis of all these genes. So late transcription means that. Then we have OP genes that are involved in DNA synthesis or replication. So now these proteins, okay, out of these, C3, N, C1, and C2 are basically involved in lysogen, and crow proteins are important for lysis. Fine. Then we have these proteins. These are important for integration. That is also involved in lysogeny also. Okay. So this is the integration site and this is the excision site. Okay. So these were the different regions present in the genome of the lambda phage. Okay. Now moving on to how this particular process happens inside the bacterial cell. So first we'll discuss about different regions present inside the bacterial cell, in the lambda genome, and what is the function of each of these sequences. Okay, so in the last slide, as we discussed, that these is involved in lysogeny. So here is the only regulation or transcriptional switch present that helps the bacteriophage lambda to decide which cycle to follow, lysis or lysogeny. Okay, so starting with the rightmost gene, that is C2. Okay, or rather than starting with the crow protein. Okay, because C3, C2, and C1 are linked, so we'll put them together. So there are five main important genes: C3, N, C1, Crow, and C2. Okay, Crow protein, Crow gene basically leads to the formation of the Crow protein, which basically represses the action of C1 and stimulate lysis. So this is important for the lytic pathway. C1 protein is first we'll go with the N protein. So N is the anti-terminator protein that prolongs the transcription after the terminators, thus allowing C3 and C2 to be produced. Okay, we'll get to know how this N functions, but in majority, like in total length, what we see, the N protein basically increases the transcription of C3 and C2 proteins, and it acts as an anti-terminator for these two proteins. Okay, then we have the C2 and C3 proteins. So C2 basically stimulates the promoter for C1 and it helps in its production, and C3 also helps C2. Actually, C3 protects C2 from proteolysis activity that helps in the production of C1. Okay, so these two proteins work in coordination to produce C1, and C1 is the basic protein that represses excision from the host and lysis of the host bacterium. So this particular protein promotes lysogeny. Okay, then we have certain promoters. So first we'll go with the PL and PR promoters. Okay, so this PR promoter is for the genes that are present on the right side, this way, and PL promoter for the leftward genes. So all these proteins present this way are for formed from PL. Okay, so PR basically helps in the formation of Cro protein, and PL helps in the formation of N protein. So that is anti-terminator. Okay, and these two like promoters are really strong promoters. Then we have certain weak promoters, that is PRM, and we have PRE. So these two are weak promoters. And these both these promoters helps in the formation of C1. Okay. So here, from here till here and from here till here, these two forms, PRE and PRM, forms the C1. So PRE stands for repressor establishment, and PRM stands for repressor maintenance. Okay, so these two promoters helps in the formation of C1 protein. And then to control these promoters, we have operators. Okay, so to control PL, we have O, the operator on the left side, that is OL. So there are three promoters, OL1, OL2, OL3, and then on the similar with the right hand side case, so we have three promoters for the OR, that is OR1, OR2, and OR3. Okay, the one operator basically is nearer to the main promoter. Okay, then we have the second and third in both the cases for promoter L and the promoter right. And the like affinity to bind to these operators also varies in this case only. So first, where anything that binds to any of these operators bind to OL or OR1, followed by OR2 and OR3. Okay, so this is how the proteins bind to the operators, and through different these two operators control the promoter PR and PL. Okay, now what happens, what proteins bind on these operators? So there are only two proteins bind to these promoters, oh sorry, operators, C1 and CRO. Okay, both have in when we have these two proteins, out of these proteins, C1 have more affinity towards operators as compared to so when both C1 and CRO are present in equal amounts, then means C1 will go and bind to the operator as compared to but when ka concentration is higher as compared to the C1 protein, so will go and bind to these promote operator sites. Okay, so here PR and PL are three operator sequence. C1 and binds to these operators with different affinities. So what will happen, talking about C1, it has different affinity for OR1, okay, as compared to OR2 and OR3. So first it will bind to OR1, then it will stimulate its binding to OR2 and OR3. Okay, that has different functions. We'll talk about it later. And OR3 has a function that it will bind to OR3 first. It has high affinity towards OR3, followed by OR2 and OR. So it's a reverse kind of thing. So first, talking about C1, OR, it binds to OR1 with higher affinity as compared to OR2 and OR3, and binds to OR3 first with higher affinity as compared to OR2 or OR1. Okay, so but no two proteins, either of these proteins, do not bind to either of the operators simultaneously. They bind to each operator one by one. If C1 is bind to OR1, then will not bind to OR3. This is the procedure. Okay, then only the basic switch happens. So these were the basic terms, and then in the last we have the terminator right and terminator left for these of the promoters, so that they can form the mRNA. Okay, So now what happens during lysogenic or lytic growth? So C1 is also called as the lambda repressor. Okay. Why repressor? Because it represses the lytic cycle. That's why it is called as a lambda repressor. And how it acts? So it has a dumbbell kind of structure. Okay. So this is a dumbbell kind of structure and it binds to the operators in dimer form. Okay. So these are dimers and in total it's a tetramer. Okay. So this is the case. Now, how it happens? So, first, when there is a lysogenic cycle, C1 will be produced. So, this is the dumbbell shape and it will act on OR1. So, it's a lambda repressor. Okay. So, it acts on OR1 and the binding on OR1 stimulates the binding on OR2. So, first, this particular thing bind on the OR1, then that facilitates the binding on OR2 and further to OR3. Okay, so this represses the PR promoter that leads to the production of crow molecule or crow protein. Okay, now when crow is in high concentration as compared to C1, okay, that means the L lytic cycle has to be followed. In that case, what will happen? This crow protein will go and bind to this particular OR3 first, followed by OR2, followed by OR1. Thus, what will happen? Oh, it will basically first re- reduce the activity of PRM followed by the PRE. Okay, so this is the case. This is how uh, so here what will happen? C1 is greater than crow, that's why it is binding to OR1 first. But when some induction happens and the phage has to shift to the lytic cycle, so here crow will be outnumbered as compared to Ci. Okay, one thing to note is Ci helps in its production as well as its degradation. Okay, so what will happen if Ci has more of the amounts in the bacteria? So what will happen? It will basically inhibits its production using the OR3. Okay, because OR3 controls PRM promoter. So it will bind to OR3 such that it will not be able to produce its in higher concentration. Okay, so this is the thing. So Ci basically acts as a repressor also, as an activator also. So when more Ci is needed for stopping the lytic cycle, so it will produce more or produce more Ci by binding to these two operators. But if it has to stop its production, then it will bind to OR3 so that PRM is also inactivated and its production also halts. That's why it is called as repressor management or maintenance. Okay, so now we'll study how uh, like uh, the cycle happens. Okay, so we'll draw it. So in the middle we have, so here is the C1 protein. On its, this is the PRM. On its right we have PR. Okay. These have three operators. One is here, the other one is starting from here and ending here, and the third one is just adjacent to that. So OR1, OR2, and this is OR3. Okay. So now, wait a second. We'll draw it. we have C3 here, followed by N. Here we had terminator. Okay, here we have promoter L, that also have three operators, OR, OL1, OL2, and OL3. Then we have C1, then we have its promoter PRM. So here we have OR3, here we have OR2, and here we have OR1, and here we have PL followed by, sorry, PR followed by Cro gene. Then here we have PRE, and here we have C2 gene, and in between them we have terminator again. Okay, so here are the five genes, and now we'll see how the things happens. Okay, PR, PL are strong promoters. Okay, so they express simultaneously and when the lambda phage enters the bacterial cell. Okay, so they starts expressing the proteins linked to them. So what will happen? PL forms N and PRE forms CRO. Oh, sorry, PR forms CRO. Okay, so now PL forming N. So N basically helps in the production of C2 and C3, and CRO helps in the reduction of C1. So it basically reduces the production of C1. So now when the lysis has to be happened. Okay, so when the conditions are favorable and the lambda phage has to do the lytic cycle. So what will happen in that case? The crow protein will be outnumbered. Okay, crow protein will be greater than n in that case. Okay, so the crow protein will go and bind to OR3 first. Okay, it will inhibit the synthesis of C1. And then what will happen if crow protein increases? Then it will outnumber N. N cannot be like, it cannot be stopped producing, but it will also be stopped by the crow protein. Okay, so it will also decrease its expression, and decreasing n's expression leads to decrease of the C2 and C3. Okay, so this is the normal scenario when lytic cycle has to be followed. Okay, so this leads to decrease in C2 and C3 levels also, so that C1 will also be decreased or it is not formed at all. Okay, now when lysogenic cycle happens. Okay, so in that case, what happens? This n will be greater than crow in that case. So now what happens? This n proteins helps in the production of C2 and C3. Okay, so now what will happen? The C2 and C3 molecules are also high. Now this C2 basically helps in the production of C1 using PRE promoter. So here RNA polymerase comes, binds PRE, and C2 is a basically activator of the RNA polymerase. So RNA polymerase cannot transcribe the C1 transcript without C2. So it will bind with RNA polymerase and allows the transcription of C1. Okay. Now C3 basically protects C2. This is C3. So from proteolysis of FTS proteins, through FTS proteins, these C3 protects the C2 molecule. So this is how they works in combination. Okay. This leads to the increase in concentration of C1 molecule. Okay. Now, when lysogeny has to be established, these C1 proteins are greater than crow proteins. Okay, so these C1 proteins and Cro proteins, as I have previously discussed, also has equal chances. C1 has more affinity towards operator molecule as compared to Cro molecule. So when both of them are in equal concentration, C1 will bind to the operator molecule as compared to Cro. The Cro has to bind the operator when C1 amount is really less. Okay, so now what happens? The C1 amount starts to increase, that outnumbers Cro. So now C1 protein will bind to OR1 and then to OR2. Okay, thus inhibiting the PR promoter so that RNA polymerase cannot bind to this particular promoter and start the synthesis of Cro. Okay, so now this thing happens, hence the Crow amount goes down in this case because it's lead to lytic cycle. So in lysogenic cycle the Crow amount decreases. Now thus C1 protein also promotes its production. Okay, that's why it initially it does not bind to OR3 promoter because it directly controls the PRM promoter. So these two promoters, PRM and PRE, helps in the production of the C1 protein. So now there is a stage that comes when CI is the amount is very high. So in that case, what happens? This C1 promoter binds to the OR3 operator also. This leads to stoppage of this particular PRM promoter, thus leading to the inhibition in synthesis of C1 protein. Same thing happens with the PRE element. So C1 promotes the proteolysis of C2 and C3 as well, so that it cannot bind to the PRE and the transcription stops. So this leads to the inhibition of the C1 molecule when it is the amount is very high in the bacteria. Okay, so this is how there is the regulation of the lytic and lysogenic cycle and how different proteins are formed as well as degraded inside the bacteria. Okay, this marks the end of the regulation of transcription in prokaryotes. Okay, so this is the end for the prokaryotic transcription, and from the next class we'll start with the eukaryotic transcription. Now we'll move on to some of the previous year questions that are asked related to the prokaryotic transcription, and we'll see how much we can cover up today, otherwise we'll move on till tomorrow. Okay, so moving on to the previous year questions, and the questions mostly are simple, just you have to apply the concept. Okay, so we have this is a DNA sequence that is ranging from 5 prime ATG CTCA to 3 prime. Okay, this is the used as a template strand, and the sequence of mRNA will be okay. So as we have discussed in the initial classes of transcription, there were two strands of DNA. This is the polarity of these strands, and out of these the RNA will be transcribed in 5 prime to 3 prime direction only. Okay, so considering this case, if the transcription has to take place, this will act as a template strand. Okay, because RNA will be formed in 5 prime to 3 prime direction. So this will be the template strand, and this will be the sense strand or non or coding strand. Okay, the RNA sequence will be same as the sense strand. So it will be complementary to the template strand. That's what we discussed in the last classes. Okay, so here the template strand is 5 prime ATG CTCA 3 prime. So the mRNA sequence will be, so what we'll do, we'll see the complementary strand sequence first. This will be T G A G C A T. Okay, so this is the coding strand sequence. This is for DNA only. Now moving on to RNA. Okay, this will turn to U G A G C A U. Okay, so now we'll see in the options which have the same sequence as the mRNA. So every time if the polarity is not mentioned in the answers, consider it to be 5 prime to 3 prime only. Okay, this is a logic behind every questions if the polarity is not mentioned in the competitive examinations. Okay, so now we'll search. So U G, so it's a, so both of the options are wrong. So U G A A G C A U. Okay, so the correct answer is option 3, right? The correct answer is option 3 because it has the same sequence as that of the coding strand. Okay, now moving on to the next option, next question. So in plant, if the sequence 5 prime GAT GGC ACG AT3 prime is transcribed using opposite strand as template, the sequence of the corresponding mRNA will be. Okay, so now in this case, so this is the template strand and this is the coding strand. Okay, so it's written that this particular sequence is transcribed using opposite strand as a template strand. Okay, so the opposite strand is used as a template strand. That means this is the coding strand. Okay, so the mRNA sequence would be same, just the T will be replaced by Q. Okay, so now GAU GG CACGAU. This will be the sequence of the mRNA because it's written in the question that this particular sequence is transcribed using opposite strand as the template. So the opposite strand will be acting as a template. That means this is the coding strand. So the mRNA sequence will be the same. Okay. So the correct option would be option B. Okay. So yes, the correct option is option B. Now moving on to the next question. Which of the following statements is not correct? So here we have to select the statements that are not correct related to any of the molecular biology processes. So we'll see these are simple statements, and we'll see everything in detail in the next few classes. Okay. So which of the following statements is not correct? First option: together with proteins, RNA provides a site for polypeptide synthesis. Okay. Then we have all DNA molecules are unbranched polymers of nucleotides. Okay. DNA is synthesized in 5 prime to 3 prime direction while RNA synthesis occurs in a 3 prime to 5 prime direction. So this is a bit tricky statement. and a tRNA anticodon may pair with more than one codon. Okay, so we'll see each of these statements. First one, when the polypeptide is formed. So this is the mRNA 5 prime to 3 prime. Here the ribosome sits. This is the small unit. This is the big unit. And what happens when the peptide bond forms? This is the uh, P A P E A site. Okay, APE site. Sorry, APE site. So here the peptide bond is transferred from this particular the amino acid to this one so that it can leave. Okay, so that peptide bond is formed by the 23S rRNA or 28S plus 5.8S rRNA. So, together with proteins, these basically combine with proteins to form ribonucleoproteins. The whole ribosome is a ribonucleoprotein, right? So, together with proteins, rRNA also provides the site for polypeptide synthesis. So, this is the case. Okay, so the first option is correct. The second option, all DNA molecules are unbranched polymers of nucleotides. Okay, this is also fine because when we study DNA, so the DNA is made up of nucleotides. So nucleotide is sugar plus base, and that is attached to the phosphate group. Now this cycle continues. The phosphate group is linked to the 3 prime OH of the sugar, sorry 5 prime of the sugar, and this is particularly linked with the base. So this is how the chain goes about. So this is unbranched only. Okay, so this is also correct. Now talking about the DNA synthesis occurs in 5 prime to 3 prime direction, while RNA synthesis occurs in 3 prime to 5 prime direction. So what happens? This is the DNA strand, and if we are talking about the replication, it happens in 5 prime to 3 prime direction always. And talking about the RNA, it also happens in the 5 prime to 3 prime direction. So this option is wrong. Talking about the fourth option, a tRNA anticodon may pair with more than one codon. Yes, this is true because the number of tRNA that are present is basically for one amino acid, that is more for like for different codons. So one amino acid can bind to different codons also. Okay, so that is why the number of a tRNA anticodon may pair with more than one codon. This is also called as wobble hypothesis. Okay, so the correct option is option 3. It's not correct. Okay, now moving on to the next question. In bacteria, heat shock response is primarily controlled by. So, as we studied in that lecture itself, the heat shock response was basically there were different sigma subunits based on the molecular weight. Okay, so the majority used is sigma 70, that is involved in many processes of the bacterial cell. The sigma 32 is basically involved in heat shock. Sigma 54 involved in nitrogen metabolism. Okay, this is how the things happen, and sigma 32 is the correct answer here because it is involved in the heat shock response proteins. Okay, so the correct answer is option 3, that is sigma 32. Then moving on to the next question. Mutations in which of the following sigma factors may be possible cause of E. coli failing to adapt in response to thermal stress? Okay, so just now we discussed sigma 70, most normal physiological processes. Then we have sigma 32, heat shock. proteins that helps the bacteria to tolerate thermal stress. Sigma 54 is for nitrogen metabolism. Okay, so sigma 32, if it's mutated, then it will, like, the E. coli will fail to adapt in thermal stress. Okay, so the correct answer is sigma 32 only, like last question. So it's the vice versa kind of question that has been asked in this paper. Okay, so answer is option 3. If uh, the question would be that the bacteria cannot be used in nitrogen metabolism due to mutation in which of the sigma factors, then that would be sigma 54. Okay, so this was the correct answer. Then moving on to the next question. Presence of sigma 70, sigma factor sigma 70 in E. coli facilitates sequence specific localization of RNA polymerase of the promoter regions of genes, phosphorylation of C terminal tail of the RNA polymerase, interaction of beta subunit of RNA polymerase with the alpha subunit, and interaction of omega subunit with the beta beta prime alpha 2 complex of the RNA polymerase. Okay, so the basic function of sigma subunit is, so as you all remember, the promoter region of the bacteria is consisting of two boxes, the -10 and -35. Okay, this is called as the Primnov box. Now, please remember the sequence of these two boxes that might be asked in any of the question. Okay, in any of the competitive examinations. Now, sigma sequence basically recognizes the promoter sequence for -35 and -10. Okay, it is linked to the RNA polymerase, that is beta, beta prime, alpha 2. So, alpha N terminal domain is linked here, and its C terminal domain is flanking out here. That leads to the upper promoter element. Okay, so now sigma subunit basically interacts or identifies the promoter region irrespective of the RNA polymerase. So if we have an experiment where RNA polymerase is without sigma protein, sigma is added alone, and RNA polymerase is not there, and RNA polymerase is there with the sigma protein. Then sigma can easily bind to the DNA molecule. It cannot bind to the DNA molecule. It will bind to the DNA molecule. Okay. So sigma protein can bind to the DNA molecule irrespective of the RNA polymerase, but it will not be able to start the transcription. Okay. So sigma protein has the role in promoter recognition. So it basically has sequence specific localization of RNA polymerase of the promoter regions of the gene. So the correct option is option 1. Okay, and the interaction between these two pro is taken care by the omega because it helps in the rearrangement of different subunits of the RNA polymerase. And phosphorylation of the C terminal tail of RNA polymerase, this basically happens in eukaryotes, not in prokaryotes, because in eukaryotes the RNA polymerase 2 has a specific tail that is phosphorylated so that it will continue to do the transcription. Okay, now moving on to the next question. Sigma subunit of E. coli RNA polymerase does not initiate the transcription and follow-up during elongation, increase affinity of the coenzyme to the promoter, binds to DNA independent of the coenzyme, ensures specificity of the transcription by interaction with the core enzyme. Okay, so just now, as I have discussed, that this region is recognized by the sigma protein. It helps in transcription initiation, leads to the formation of open complex, and it basically leaves the RNA polymerase during elongation. Okay, so here the sigma protein falls off, then only the elongation or uh, the basic polymerization of the transcription takes place. Okay, so it initiate transcription and follow up during elongation. Yes, it increase affinity of the coenzyme to the promoter. Yes, because the RNA polymerase cannot bind to the promoter directly, so it increases the affinity of RNA polymerase to the promoter. It binds to DNA independent of the coenzyme. Okay, it ensures specificity of transcription by interaction with the coenzyme. So it basically cannot bind to the DNA independent of the core enzyme. Okay, so it will bind only in the presence of RNA polymerase because it cannot initiate the transcription on its own. So only the binding procedure can be done by the sigma, but the transcription has to be initiated with the help of RNA polymerase. Okay, so the correct answer is option 3. It can bind to the DNA, but it cannot bind to the DNA independent of the core enzyme. Okay, so moving on to the next question. One of the mechanisms used by bacteria for adaptation to changed environment is altering transcription of their genes. In this regard, which of the following responses is not found in bacteria? Okay, so the bacteria, when adapted to change environment, alters its transcription of their genes. So, which of the following responses is not found in bacteria? A gene with two different promoters for expression in different conditions. Okay. Use of different sigma factors for transcription of genes. Yes, this is present. For example, just now we saw about sigma 32 and sigma 54 that is involved in different transcription of different genes involved in different functions. Expression of alternate alpha beta and beta 2 beta prime subunits and expression of anti sigma factors. Okay, so the correct answer is option 3. So here the gene can be linked to different promoters also, but it cannot help in the activation or expression of alternate alpha beta and beta prime subunit because they are same in whole of the bacteria. Okay, so everything is synthesized from this particular subunits only, forming the core enzyme. So the correct option, this does not happen in bacterial cell. Okay, anti sigma factors can be formed, and these two also can be done in bacteria, but this particular thing, formation of alpha beta and beta prime subunits alternative, cannot be formed in bacteria. Okay, so moving on, this was the last question for today, and then we'll see more questions about the operon system tomorrow, and tomorrow we'll be also starting with the transcription in eukaryotes. Okay, so stay tuned and see you all guys tomorrow.